so we'll move straight to the flash talk. I'm going to read their profiles and then we invite them over. So Mr. Kadir O, he graduated with a BS in biotechnology from James Madison University. Following this, he spent 2 years as a research assistant at Thomas Jefferson University, where he studied amyotrophic lateral sclerosis. Kadir is currently a PhD candidate at Purdue University, where he studies the structures of protein aggregates in neurodegenerative diseases. So please, can we all welcome Kadir O to take his session? Thank you.
Thanks for the introduction.
You're welcome.
All right, well, I'll get started. All right, so I'll tell you guys today a little bit about structural studies of prion protein filaments from Gerstmann-Straussler-Scheinker disease. Let me close this real quick. But first, a little bit of background. Back in 1750, there was a nobleman called Johann Leopold, and he noticed that there were sheep that were scraping themselves against fences, and they would, over time, they would stop eating, they would lose their appetite, they would get very sick, and then die after a while. So this was called scrapie disease. Later in 1920, two German physicians, Creutzfeldt and Jakob, independently described a patient who was 23 years old that had neurodegenerative symptoms similar to Alzheimer's disease, but in such a young patient, where she started to walk very abnormally. She also lost her appetite. She began to develop tremors and seizures. And within months, the patient passed away. And when they looked at the brain tissue, they saw lots of lesions inside of the brain, or holes inside of it. Later in 1962, a similar disease emerged called Kuru in Papua New Guinea. And what they saw was, in the same patients, similar symptoms such as loss of appetite and death within months. And inside their brains, they see the same holes or lesions. So similarities between these diseases. In 1965, a Nobel laureate, Carleton Gajdusek, thought that these diseases were potentially being caused by a type of virus, except this virus had potentially unusual biological properties. But when they used UV inactivation on these viruses, they saw that the infection actually was not caused by a virus. And lastly, very recently, and something you might be familiar with, is mad cow disease. And these cows actually exhibited very similar symptoms to the scrapie sheep. And when you look at their brains, you have the same holes inside the brain, the spongiform lesions. So all these diseases seem to have a very common kind of unifying factor. So what is the link between these diseases? So in the 1990s, Dr. Prusner did several experiments where he was able to isolate the disease agent from the sheep and the cows and inject them into hamsters. And when they injected these filaments, these protein aggregates into the hamster brain, they died within months and developed the same lesions in the brain, these holes. So he actually concluded that it was actually a protein infection, not a virus or a bacteria that was killing these individuals and these animals. And so these protein aggregates, known as amyloids, are present in multiple neurodegenerative diseases, including Alzheimer's, Parkinson's, Lou Gehrig's, and prion protein diseases. And they're characterized by memory loss and generally rapid death within months to years. So on the left is a healthy brain, and on the right is a atrophied brain from the disease process. So these prions, or these aggregates, usually derive from a native protein, so it has some kind of structure, and they can become misfolded and turn into aggregates. These aggregates are known as amyloid filaments. And in these amyloid filaments, the proteins are separated by 4.8 angstroms. So each of these ribbons represents 1 unit of protein. And these filaments are very resistant to proteases, so they can't be cleared out of the brain very easily. And so in the case of many neurodegenerative diseases, we see different proteins form amyloids in different diseases. And each protein actually can take on many different forms of this aggregate. Here you can see that tau protein in all these different diseases can take on many different folds. So one protein structure doesn't have one fold in the case of amyloid proteins. So the question is, why do we care about these structures? Well, diagnosis is very difficult for these patients. So using things like fluorotazapine, which is a PET tracer, you can light up regions of the brain that have these aggregates. So you can see control patient doesn't have any lit up, whereas the Alzheimer's disease patient has lots of the areas lit up with this diagnostic ligand. Also, if we know the structures of these filaments, we can potentially stop them from forming or disaggregate them, which could potentially lead to slower neurodegenerative disease or potentially curing the disease altogether. So today I'll tell you a quick story about Gerstmann-Straussler-Scheinker disease, which is a prion protein amyloidosis with both tau and prion amyloids. The symptoms of this disease are very similar to Alzheimer's in that there's language impairment and motor dysfunction, and later in the disease, there's dementia with brain matter loss and death. And you can see here a healthy brain with just full-size brain, normal neurons. But in the case of both Alzheimer's disease and in Gerstmann-Straussler-Scheinker disease, inside the cell you have tau protein, which forms neurofibrillary tangles. And outside the cell, you have amyloid plaques made of prion protein. It is thought that the tau protein binds along the microtubules and helps for the transport of mitochondria and other things like that. But when tau becomes hyperphosphorylated, it can fall off of the microtubule and destabilize it. And this tau that has come off of the microtubule can then form amyloid filaments and aggregates. In the case of prion protein, it sits on the outside of the cell, and part of it can be cleaved off. And this released protein can actually also form amyloid aggregates. So both tau and prion protein form these amyloids. When we look at a slice of the brain, you can see here, stained in brown, are the prion protein aggregates used in an antibody. When we zoom in even closer, we see the same prion protein aggregates in brown, which are outside the cell, and labeled in purple are the tau filaments within the cells. Zooming in even closer with an electron microscope, you can see the tau inside of the cells forms these long string-like aggregates, and the prion plaques outside the cell form more of these radial aggregates forming from a center and branching out. So in this disease, both tau and prion form amyloid filaments and aggregates inside and outside of the cell. So the question is, what is the structure of these amyloids? So recent work from our group, we saw the structure of the tau filaments from within the cells, and we found that the structure is identical to the tau found in Alzheimer's disease. The question is, what is the structure of the prion amyloids from this disease? So with our collaborators from Indiana University, they sent us 2 patient brain samples, one with homozygous at position 129 for valine, and one heterozygous at position 129. Something to note is that the valine 129 accelerates and predisposes individuals to prion diseases. So when they sent us the sample, it was highly aggregated. You can see this is an aggregate of actually filaments. When you zoom in closer, you can see these rod-like structures. So these are the individual amyloids that make up the plaques. But we can't study these by Cryo EM, these large plaques. We need individual filaments. So we use a disaggregation protocol where we vortex the sample and allow it to pellet slowly and take from the top portion where it's less aggregated. And using some detergents and sonication, which is high power sound waves, we're able to break apart the filaments. So right after this, you can go into the negative stain or plunge freezing used for Cryo EM. So after this procedure, we see the filaments are very well separated and individual, which is good for Cryo EM. But when we freeze the protein, we found that the protein was stuck to the edges of the holes instead of inside the holes where we want to image them. So we used graphene oxide, which is a paper-thin layer of carbon, to layer over the Cryo EM holes so that the protein could stick to it. Once we did this, the prion filaments stuck inside of the holes where they can be imaged. So after a lot of processing, which I'm not going to show you, we found 2 types of filaments containing 2 individual strands that wrap around each other. You can see that they're both identical to one another. However, they're flipped by 180 degrees or rotated. The other one also had the similar structure. However, this one was inverted. You can see that the same protofilament structure exists in both types. So when you take a zoomed in look and you do model building or atomic model building, you can see each of the protofilaments consist of individual layers of protein that are identical to one another. And it's on the other side, it's the same protein, but rotated 180 degrees. When we look at this, it's actually a very flat layer of protein that stacks on top of one another, as you can see over here. And so there's a lot of stabilizing features in here. You can see these side chains alternate with one another, which is a steric zipper, which makes a very stable structure and prevents the protease degradation. And you can see also a very densely packed hydrophobic pore here that helps the protein folding. And also there's a patch of positively charged residues, lysines, that can potentially attract other proteins or cofactors. And lastly, this bulky phenylalanine residue kind of closes the door on the structure. When we compare the two structures, again, we see each of these units is identical to one another for type 1 and type 2, except in type 1, it's rotated 180 degrees, and in type 2, it's kind of a mirror image where one filament is going the other direction. When you look at the structure, there's lots of beta strands, which engage in hydrogen bonding between the layers. There's actually 9 total. And when we look at the structure, you can see all these beta strands with these yellow dashed lines engage in this hydrogen bonding all the way down the length of the amyloid that makes it very stable. In addition to this, we saw that our protein backbone had lots of extra densities near it. These extra densities were concentrated around the positive charge residues, and some were inside of this hydrophobic core. The thought in the amyloid field is that these extra densities might be non-protein agents such as ions or DNA or RNA or lipids that may be molding the conformation of these amyloids. And lastly, remember back to the valine 129, which accelerates prion disease. In both patients, we saw that the density at this position, valine 129, was consistent with valine, even though patient 2 had methionine and valine. So when you do a brief simulation, you can see that methionine prion protein could not be incorporated into this region because it would induce steric clashes. So this shows how valine 129 prion protein can be incorporated into prion filaments, whereas methionine cannot. So some conclusions from this are that prion protein amyloids from this disease have a common subunit, but with different forms of packing, and that the 129 methionine prion protein is not incorporated into prion filaments, which is a good reason for why 129V accelerates these diseases. So some of the future directions include studying other prion diseases and comparing them to this structure, and as well as taking recombinant prion protein and forming filaments from these in an in vitro setting and comparing them to the patient structures. We can see that prion structures from the disease states, so this is my structure from the human, and this is a structure from a hamster, are very different from one another. So the same protein forms very different structures. And many of the in vitro, so just in a test tube with recombinant protein, also have very different folds. So this kind of asks the question, what are the factors that influence these variations? So there's quite a few, I'm not going to go into depth, but things like mutations in the sequence, truncation of the protein from full length to smaller proteins, and lots of things like post-translational modifications, the chemical environment, such as pH, salts, the oxidative state, and even cofactors such as metals and ions and other things can influence these variable folds. So general future directions for the field will be to take these recombinant proteins in a test tube setting and try to figure out what causes them to fold into the correct fold that we see from the human brain. Other things we can do are assess the amyloids formed in animal models. And lastly, a direction that's still being explored is that we need to develop more diagnostic and therapeutics for these diseases, since they're currently incurable. With that, I'd like to acknowledge my lab members and just Hockmeyer and Purdue Biological Sciences, and I will take questions. Thank you.
Thank you so much, Kabir, for that very explanatory presentation. I guess it made us see the practicality, the application of how we can use Cryo EM to solve problems, in our world today. So please, can we have questions? Can we have questions? You can either raise your hand or type your question in the chat box, please. Any questions? Mr. Olade, would you have a question? All right. Comments are coming in the chat box. Thanks so much for the presentation. It was clear and well understood. All right, then.
Thank you.
Yes, it was clear and well understood. Okay, can we have more comments? Can we thank our facilitator, Kabir, for the presentation? It's very interesting.
Hello, I don't know if they can hear
I can
me.
hear you, ma'am.
Yeah, this is the peak of this workshop for me, right from the morning session, the afternoon session to now. I wanted the application. Where is the application of this scientific technology? And it's a good presentation. At least we now know where this technology is going to and where it can be applied in the future. Thank you so much for the presentation. Thank you, the facilitators, and thank you, Jaris, the director. I am grateful. Thank you so much. Yeah.
Thank you. Appreciate the kind words.
Thank you so much, Dr. Akinsomi. We appreciate you. Okay. Others are saying thank you for the presentation in the chat box. All right, thank you so much, Kabir.
Thank you.
Yes, thank you so much for the efforts for putting this together. Thank you for your time as well. So we'll be moving on to the next session. The next flash talk is going to be taken by Ms. Shubham Dubey, and she's going to be